Right, hello. It's the summer holidays, and I have just finished my GCSEs, and I'm hoping to be taking A levels in September. Um, and so, in preparation for that, I've started a series of videos which will be introducing the key ideas of A level and AS biology, um, to try and prepare me and any of you for the course starting in September, or whenever you'll be doing it. Okay, so firstly, we're going to be talking about proteins for this video. Um, so you surely know that proteins are made up of long chains of amino acids, right? Um, and that there are 20 different amino acids used in proteins. 20 different amino acids, that's quite important. You've got to remember that there's 20 different amino acids that can make up proteins, basically. Um, they all have a structure of NH2, RCH, COOH, where the R is one of 20 different chemical groups, and that is what determines which of the 20 amino acids the amino acid is, which links to earlier the fact that there are 20 amino acids. It's all based on the chemical group of the R group, also known as the side chain, as we can see in this little diagram, top right. And you also have the amino group, which is the NH2 part, and the carboxyl group, which is the COOH part. All amino acids contain carbon, oxygen, hydrogen, and nitrogen, and some of them contain sulfur. So you can see up here, you've got hydrogen, nitrogen, carbon, and oxygen. There's no sulfur in this one, but they can, some of them at least, contain sulfur. Chains of amino acids in proteins are held together by strong peptide bonds. A peptide bond is a chemical bond formed between two molecules when the carboxyl group of one molecule reacts with the amino group of the other molecule, releasing a molecule of water, or H2O, and leaving the two molecules to be joined together. This is called a dehydration synthesis reaction, or is also known as a condensation reaction, and it usually occurs between amino acids. So, for example, this is an amino acid, obviously, and if it was to join with another one, then a peptide bond would form between two of them, and H2O would be given off to allow that bond to form. And proteins can be up to several hundred amino acids long. So there can be lots of amino acids all joined together with peptide bonds in a chain, a long chain, in a protein. The order of amino acids in a protein is what determines its structure, and the protein's structure is what determines how it works. The order in which the amino acids are arranged in a protein chain is called the primary structure of a protein. So just how the amino acids are like the order they're in, just the order they're in the chain. That's its primary structure. Some protein chains can coil up or fold into pleats. Pleats are just like folds, basically. If you imagine a piece of cloth just folded, it would be pleated. That are held together by weak forces of chemical attraction called hydrogen bonds. The coils and pleats are the secondary structure of a protein. So basically, in the secondary structure, it's just all the coils or the pleats, to keep them actually together, and in that structure, hydrogen bonds hold them together. And then the tertiary structure is where some proteins, and especially enzymes for this one, well, they have a tertiary structure, and that's where the coiled up chain of amino acids is folded into a ball, which is held together by a mixture of weak chemical bonds, such as hydrogen bonds, and also stronger bonds, like disulfide bonds. And I'll show you some examples of that in a bit to make it clearer, because the whole structure bit is quite confusing. So I'll show you what I mean by what I've just talked about in a minute. if the protein has a roughly spherical shape, it's called a globular protein. For example, enzymes are classed as globular proteins, and we'll also look at an example of that later. Okay. Right, so this is just to visualize the 20 amino acids. You won't need to memorize all these at the moment, but it just shows you the 20 different ones, and you can see where I've circled it in red. I've circled the amino group, so the NH part of each amino acid, as you can see in each one. Where I've circled in yellow, I've circled the carboxyl group, so the COO part, or COOH, but this diagram's a little weird about how it presents the amino acid, but it's the same thing. And then at the bottom, in green, the R group has been circled, and so the R group is different in every single one, and that's what determines the amino acid that it is. So each one's different, so it's going to be a different amino acid. For example, if this one, glycine, if its R group was CH3, it wouldn't be glycine, it'd be alanine, and vice versa. So glycine is glycine because it has H, or hydrogen, only in its R group. So that's how they vary, and that's what makes amino acids different. Important little thing to remember, which will be useful in understanding how they're bonded this way as well, is that hydrogen can only form single bonds, whereas oxygen can form up to double bonds, nitrogen can form up to triple bonds, and carbon can form 4 bonds. If you remember from GCSE, diamond, every carbon atom in diamond was bonded to 4 other carbons. So but it's quite easy to remember that. Okay, so this is the structure part again. This is the quite confusing bit, but basically the primary structure is just the way they're organised, like laid out. So you can see it's just the order the amino acids are in. So you can see these are all different amino acids, right? Because they're all different colours. The ones that are the same colours are the same amino acids, and the way they're laid out, that's the primary structure of the protein. The secondary protein structure is, you can see it's folded here or coiled here, and these folds and coils, or pleats for folds, they're held together by strong chemical forces of attraction as hydrogen bonds, which like hold them together. Tertiary structure is where the different folds and coils within the amino acids are bonded with not bonded, just attracted with the with other folds or coils, and this can cause it to form what's like a ball shape here. And finally we have the quaternary protein structure, which is a protein consisting of more than one amino acid chain. That's pretty self-explanatory. It's a protein with more than one amino acid chain. Here's an example of a globular protein. It's the enzyme catalase, and if you remember earlier we said that enzymes are an example of globular proteins, and you can see it's got that spherical shape, which makes it globular. So that's just an example of a globular protein. And then finally you can see what happens when you combine 2 amino acids. So here we have amino acid here and amino acid here. We can see that water, or H2O, is given off, and what actually happens is the bond here joins with the N here. Doesn't join with the H though, because you remember hydrogen can only form 1 bond, so it has to join with the N, and that connects that like that. And you can see that carbon can form 4 bonds, so that's fine, and nitrogen can form 3, so it all works. Whereas if we were to if it was to bond with H, well, that just wouldn't work because hydrogen can only form one bond. Same here. We have an amino acid here and an amino acid here. Water is given off, or H2O, and a peptide bond is formed. This is the peptide bond in this one, by the way. The peptide bond is formed between the two amino acids, and because carbon can form 4 bonds, this one works fine, and nitrogen can form 3, so this works fine. Hydrogen can form 1, so that's fine, and oxygen can form 2. So you've got to make sure that each atom has the right number of bonds to make it work. Otherwise, you've probably done something wrong. But yeah, it's just a way of showing how it works. Same here, peptide bond forms, H2O given off, and you can see each atom has the correct number of bonds. Again, when two amino acids react, or rather join together, it's caused by a condensation reaction between the two amino acids, where the amino group reacts with the carboxyl group, water is given off, the peptide bond forms. And then finally, here's another example down here. You can see the same thing going on, and same here. So I hope that was helpful. That's just a brief introduction to proteins as part of the A-level or AS level syllabus, and I hope that helps you understand them a bit more in preparation for the A-level course in September. Thank you for watching. Goodbye.